I've had a number of people ask us just how we go about making these various models that we produce using 3D printing technology. Most of these are models that we build of different molecules. Sometimes they're more complex protein structures. What I'm going to do today as part of this tutorial is take people through some of the simpler molecules that we build. Usually I like to do a project if I'm going to go through and start something new. So this project that I'm working on currently is an effort to kind of build modular components to be able to construct a DNA helix. And so that's what we're going to go through. The first thing you have to do here, if you're going to build something, is you've got to have some kind of coordinate file system. The files that I use here, I'm going to get from the protein data bank. You can get your molecule coordinates from other software, I'm sure, but at the end of the day, I usually need to be working in some kind of PDB file format. So I've gone to the protein data bank and I've found a specific DNA helix that I'm going to use here to build my model. There's an identifier over here, 476D. I'm going to kind of pay attention to that, but this is just an oligomer that somebody has constructed and gotten a crystal structure for. The first thing we've got to do is we've got to get this into some type of 3D rendering program so we can do some manipulations, because we don't want to print the entire thing all at once. So we want to go over here and find the download files and pick up PDB file. We want a PDB text file for the program that I'll be working in. We'll click on that and we'll save this. I'm going to actually save this on just on my hard drive here under a folder called structures, and you can see I've already actually done something similar to this previously, so we will just go ahead and hit save, replace the current version, just because I'm going to go through this again just to show how this is done. Once it's done, you can also click on open. And for the next step, you're going to have, as I mentioned, some kind of 3D rendering program that you're going to use to do the manipulations to the molecule. I work in Viewer Pro. You can work in other software, and you can try to get yourself a version of Viewer Pro. Viewer Pro was free up until a few years ago, but somebody's purchased that up now, and I think you have to at least give them your email if you're going to use it for various purposes. There's other software programs you can use besides that, plenty that are available. Protein Explorer, some of these other ones. I'm just working in Viewer Pro because it's what I'm most comfortable with, so I'll do my descriptions here. But the biggest thing is we've got way too much stuff here to do a print of. I want to kind of break this down into a simple module of just a base pair unit that I can then build up from. So one thing I'm going to do is go under View, and I'm going to click on Hierarchy, and then I'm going to get rid of some things. I don't really intend to print out all the waters that are shown in this molecule. We're just going to throw those out. We're also going to throw out the calcium ions that are shown here as well. I don't intend to print them either. There's some other calciums that are associated with the other strand. What I am going to do is I am going to go through and just pick 3 different bases. In this example, I'm going to pick out an AT base pair. So I'm going to come down here until I get to an AT base pair. And what I'm going to do is leave a flanking base pair on either side of that. So I'm just going to click and select the base pairs I don't really want to manipulate and work with. You can see there's some issues here, especially on the ends where you really don't have base pairing, just because that's how the crystal formed when they got the structure. And so there's some things that really aren't part of the DNA helix that we're trying to work with here. We're going to delete all of these. We're going to come to the other complementary strand, and we're going to find here the complementary bases, which are those 3. And we're not going to get rid of those, but we're going to get rid of everything else because we're just trying to simplify this structure at this point in time. So we delete all that. We now have the 2 bases that we're actually concerned about and want to actually do some manipulations with. and get into our 3D rendering software so we can make our end goal, which is to get an STL file out of these. So I'm going to do a couple things here really quickly. One is I am going to center this molecule, and I'm also going to blow it up a little bit so that I can see it. We'll rotate around. Like I said, I can see the different base pairs. I'm going to kind of manipulate this just so I can orient myself. Again, because this is DNA, we've got the actual base here in the center. It's actually forming the planar base pair between the two. This should be one of the it should be AT. That's the two pieces here, the A and the T that we'll be working with. And I can see where the oxygen is here in the ribose unit. This then, based on the conventional numbering, it's going to be carbon 1, 2, 3, 4. Over here is 5. Then I've got my phosphates and my backbone. And because I'm going to try to build something modular, I'm going to cut out most of the other pieces of the DNA of the bases except for the carbons. I'm going to leave the carbons here now because I haven't quite decided how I'm going to try to piece these together yet. I want to be able to build these one unit at a time, so I've got to kind of decide that. I'll probably decide that actually once I'm in my final program. So for right now, I'm just going to cut out most of the remaining parts of the molecule that I really don't want to use for this module. So I'm going to leave myself the carbon that's attached to the phosphate up here. Erase everything else. And I'm going to leave the carbon that's attached to the phosphate down here. Erase everything else. I'm going to flip and orient this again. Do something similar on this side. Leave the carbon. Get rid of the rest of the molecule. Leave the carbon. Get rid of the rest of the molecule. So now I have my base pair, the riboses, the phosphates, and a carbon that's attached up on either side, the flanking carbons that are there. And we'll manipulate that later when we get into the other programs. But for right now, we're just about there. For this model, I actually want to include my hydrogens. Some of your software program may have already included that. This one kind of makes you select it. And so I'm going to add the hydrogens in just so I can come in here again and see my hydrogen bonds, the two hydrogen bonds that I'm interested in here for the AT base pair. Okay, so as I put some more thought into this, I really do want to build a module. So I actually want to start thinking about this as if I insert various modules on top of each other to make this. What pieces need to be conserved? So you got to think of now at this point in terms of the DNA. We know that this group right here, that phosphate from this unit, is going to be also the same phosphate up here. So really what we're looking at up here is the 3 carbon from the ribose that's up above this. This again is our 5 carbon, and this is our 3 carbon down here. So the piece that I really want to have, I want to be able to know how the stick is going to go into the carbon down here. I'm going to build this as a ball and stick model, and so I'll actually have a carbon ball here and a place to put a peg in down here. So that's the piece I need to conserve. So I'm actually going to remove a few more things off of this. Now that I've drawn in my hydrogens, this will make it a little easier for me to see this. I'm going to get rid of that. So now I've left myself with just the stick where the phosphate was at. And then up here, I'm going to leave the stick, the phosphate as well. But I'm going to get rid of the hydrogens here. because those will just be things I'd have to erase in my next software program. So that actually leaves me the stick that would be going into the carbon down here, and the stick that is from the previous one going in the carbon down here. I'm going to flip this thing around, do the same thing on the other side. I'm going to delete the hydrogens there off of that carbon, and then going to delete that, leaving myself with a phosphate there. That allows me to actually just have the pieces of the module, which is what I'm looking for. So this is really all I want to have here. Now at this point, I'm going to save this. I'm going to save it as DNA base pairs AT476 with the designator here. And I'm going to click save. That should be all the pieces that we're going to want to take into our next software program.